Today, I will discuss a topic from the subject immunology, and the topic is innate immunity. But before moving to innate immunity, we first know about what is immune system and what is immunity. So, in this slide, I am going to discuss about immune system and immunity. First, immune system. Immune system is a dynamic network that comprises cells and proteins that help to defend organisms from foreign invaders and cancer. Now, let me explain what is the meaning of foreign invaders here. Foreign invaders are generally known as antigens in the term of immunology. These foreign invaders can be virus, bacteria, or any other kind of pathogen like your protozoan. Now, certain times our normal cells stop functioning normally and they change or transform in some kind of cancerous cells, and this give rise to the disease cancer. Now, in that case, our immune system also functions to eliminate these transformed cells so that body will remain in its normal system. Immunity is the state of a living system. We can say immunity is the property of immune system. It is the state of any living organism in which it tries to protect itself from antigens or harmful entities that are foreign to that organism. All unwanted changes that occur spontaneously in a system itself, like transformation of normal cells into cancerous cells, this is also a property against which our immunity fights to sustain all the normal functioning of a living organism. Each and every organism has some kind of immunity for defending itself. This line is an example. I can explain you with an example like a normal single cell protozoa, or we can say a normal single cell prokaryotic organism. They all have their immunity. For a very short example, restriction endonuclease present in bacterial cell is the enzyme that fights against viruses. That enzyme breaks the DNA of viruses. So similarly, if we talk about higher organisms like plants also have certain kind of immunity, which is due to secondary metabolites like flavonoids, alkaloids, phenolic compounds. These all provide the immunity to plant body. Similarly, in case of humans or vertebrates, we have T cells, we have B cells, macrophages, natural killer cells, eosinophil, basophil. These all kind of cells help to protect our system and provide immunity. Now, before going to innate immunity, we should know about there are two basic components of immune system. The two basic components include innate immunity and adaptive immunity. Innate immunity, the word innate means by birth or inborn. That is, we are born with such kind of immunity. It is a primitive immune system that is present in all living organisms, whether they are vertebrates, whether they are invertebrates, or whether they are prokaryotes. Second one is adaptive immunity. Adaptive immunity is that kind of immunity which we encounter after the infection of microorganisms. In this kind of immunity, there is always a memory response. That means a memory develops for a particular pathogen. But in innate immunity, no such kind of property is there. We don't have any memory for a particular pathogen. So now moving to the innate immunity. Now we will discuss about properties of innate immunity. First is less specific with broad reactivity. Now what this means? Less specific means it is not specific for virus, bacteria, or any other pathogen. It functions or it protects our body from viruses, bacteria, protozoan in the similar manner and with broad reactivity, means it recognizes a whole pattern of biomolecules. It is also known as first line of defense, which fight against infection and present before the onset of infection. Second, set of disease resistance mechanism not specific to a particular pathogen. Again, less specific, I have defined it that it is not specific for any particular kind of pathogen or a particular pathogenic species. Includes cellular and molecular components that recognize pattern of biomolecule. I can give you an example of this line. Toll-like receptor 2. It is a molecule that only recognizes lipopolysaccharide present in gram-negative bacteria. Toll-like receptor 2 will never recognize lipopolysaccharide present in gram-positive bacteria or any other or in any other form. That means the molecular components always recognize a particular pattern of biomolecules. There are a number of gram-negative bacteria with lipopolysaccharide, but they are recognizing a specific pattern of lipopolysaccharide. Innate immunity comprises phagocytic cells. Phagocytic cells are those cells that engulf the foreign particles or antigens. Phagocytic cells involve blood monocytes, macrophages, natural killer cells, etc. Dendritic cells are also the example of such kind of phagocytic cells. Skin, the first barrier. Chemokines, again the molecular components of innate immunity. They play important role in innate immunity and protect our body from foreign invaders. The last is uniform in all members of a species. That means innate immunity are those properties or those immunities that are specific for a species. All human beings have same kind of innate immunity, but acquired immunity can be different. For example, maybe I have acquired immunity for virus A and somebody else has an acquired immunity for virus B. So, but if it is innate immunity, we both are protected for virus A and virus B. I think this difference may be clear. Now moving to the next slide. Innate immunity is divided into 4 attributes. The first attribute is anatomical barrier. The second is physiological barrier. Third one is phagocytic barrier, and fourth is inflammatory barrier. Now, this is a table which shows the 4 attributes along with their function mechanisms. Anatomical barrier include skin and mucous membrane. Physiological barrier include temperature, pH condition of the body, and chemical mediators. For example, lysozyme. In our eyes, we have tear glands. These tear glands produces a particular enzyme known as lysozyme. These lysozymes function against the bacterial cell wall. Lysozyme works on bacterial cell wall, breaks the bacterial cell wall, and thus harm or thus stop the activity of bacteria in our body. Phagocytic or endocytic barriers. Phagocytic cells like monocytes, etc., they work on these kind of barriers. Monocytes, neutrophils, tissue macrophages all comes under phagocytic barrier. Inflammatory barrier. We have generally heard the term inflammation. We always say we have swelling and it is inflamed. The area which is swelled is called inflamed. When we have any kind of injury, when we have any kind of infection, in those conditions, the system or immune system works and produces inflammation. In that condition, there is redness, there is swelling, there is pain, and there is heat also in that area. This collaborative function of immune system is known as inflammation, and we study it under inflammatory barrier. Now moving to the anatomic barrier, or it is also known as physical barrier. Skin, the first line of defense. Now we will discuss about skin. Here you can see the image of skin. Skin consists of epidermis, dermis, and below the dermis there is fatty tissue, or we can say it adipose tissue. Epidermis is the uppermost layer which is made of epithelial cells. The topmost epithelial cells are dead and have a protein known as keratin. This protein provides a waxy texture to our skin. Now, moving downwards in dermis, dermis have hair follicles, dermis have oil glands, dermis have sweat glands. These oil glands are also known as sebaceous glands, and its secretion is known as sebum. In epidermis, there you can see melanocytes. Melanocytes are the cells that produces a chemical known as melanin, which impart skin color or color to our skin. You can here see that blood vessels are also present in the region of dermis. I would like to discuss a point about sebum. Sebum is a substance which contains lactic acids and fatty acids, and sebum provides a pH between 3 to 5 to our skin, and that pH is not normal for the survival of microorganisms. So skin provides a pH which helps to eliminate the microorganism from our skin in normal conditions. If the person is not have any wound or any kind of further infections or any kind of allergic response. Now, moving to the physiological barrier. Physiological barrier comprises temperature, pH, some soluble proteins. Temperature. First, we will discuss temperature. Temperature of normal human body ranges from 97 to 99 Fahrenheit. And similarly, for example, in chickens, this temperature is 105 to 107 Fahrenheit. Now, have you ever heard about anthrax? Anthrax is a disease, very fatal disease. So, chicken has a body temperature of 105 to 107. This temperature resists chickens from having anthrax, but humans don't have such efficiency that they can fight against anthrax in their normal body temperature condition. Now, pH. Our stomach consists some gastric acid. This is an innate physiological barrier. When we ingest food, when we take food, when we take water, or we inhale air, some microorganisms always move inside our stomach. But the low pH of our gastric enzymes and HCl present in our stomach help to eliminate these microorganisms. Soluble proteins like lysozymes, interferons, and complement proteins, they are very helpful as a physiological barrier. Our eyes have lysozyme. Interferon, as the term suggests, there is some kind of protein that interferes. When we have a cut or a wound in our skin or any kind of lesion or abrasion in our skin, and there is a viral infection, when our skin is getting virally infected, then these interferon help to fight against those virus particles. Now, complement protein, another system. Complement proteins and complement system is part of both innate as well as acquired immunity. Sometimes some proteins function in innate immunity, and some proteins and some kind of complement system function in acquired immunity also. When there is dependence of antibody, the complement system shifts towards acquired immunity. When there is no need of antibody and it fights against the microorganism without consideration of antibody, then the complement system is a part of innate immunity. Lysozymes are hydrolytic enzymes. Hydrolytic enzymes, they function by hydrolyzing bacterial cell wall. Interferons comprise a group of proteins produced by virus-infected cells to fight against the virus particles. And complement proteins are a group of serum proteins that are present in serum and mostly are in inactive state. They only get activated when they encounter any kind of pathogen or antigen. Here we can see a diagram which represents physiological barriers. Salt from perspiration. While we perspire, we breathe in, out. Some salts are also deposited in our nasal tract. Lysozymes degrade peptidoglycans, that is your LPS layer of bacteria. Peroxidase enzyme that decompose H2O2. Lactoferrin that binds iron particle. Defensin form pores in microbial membrane. Defensin is also a kind of chemokine. It also binds and forms the pore in microbial membrane so that there is a salt imbalance in microorganisms. Acids, low pH in sweat or in stomach. That means low pH of skin as well as in stomach help to fight against the microorganism in physiological manner. Now, phagocytic barrier. Phagocytic barrier. This mechanism of innate defense is based on ingestion of extracellular invaders by phagocytosis process. When a cell engulfs a whole particle or eats a whole particle, engulf means eating. When a cell eats the whole particle, then this is known as endocytosis. But if the cell is engulfing or eating something in liquid form or a toxin or any kind of digestible proteinaceous form, then this process is known as pinocytosis. Phagocytosis or pinocytosis. Phagocytosis and endocytosis both are the same terms. In phagocytosis, phagocytes or phagocytic cells like blood monocyte, neutrophil, tissue macrophages engulf pathogen and forms a vesicle known as phagosomes. This process is mostly receptor mediated. Now, I will explain it through a diagram. Here we can see PAMPs, PAMPs, peptidoglycan in a cell wall. This is a microorganism. Here it is going to adhere with the help of TLR on macrophage. Now, there is a chemotaxis and adherence of microbe to phagocytic cells, that is your macrophage. Chemotaxis, this term means attraction through chemical, a chemical attraction between two different cells, bacterial cell and the macrophages. After chemotaxis, after this attraction process, the bacteria get adhered at the phagocytic cell. Now, this phagocytic cell ingests the microorganism through the process of phagocytosis. In third step, here we can see in third step, there is the formation of vesicle. Here's a round vesicle. This vesicle is known as phagosome or phagocytic vesicle. Now, there is another vesicle already present in the macrophage that is known as lysosome. And the lysosome contains lysozyme, the enzyme that degrades bacterial cell wall and kills the bacteria. This is also known as digestive enzyme of the cell. Now, lysosome and phagosome fuse together. They fuse together through vesicle attachment and forms a second structure that is known as phagolysosome. This phagolysosome is also known as early phagolysosome. Now, in fourth step, when there is formation of phagolysosome, again, partial digestion of microorganism starts taking place in phagolysosome. Indigestible material, that is the bacterial cell that remains indigested or residual bodies are thrown out through macrophage. Now, let's discuss about something, how this works, how this lysozyme works. So, the lysozyme has a property that it is acidic in nature. Here I have told you about early phagolysosome. Early phagolysosomes have pH between 6.8 to 6.1, whereas late phagolysosome has a pH of 4.5, which is highly acidic in nature. So in this way, the phagocytic barrier functions and kills the microorganism. Moving towards the last and fourth inflammatory barrier. First, we will discuss about this, then I will show you the diagram. Inflammation, tissue damage. First is release of vasoactive and chemotactic mediators like histamine, serotonin, etc. Second is vasodilation. This process increases the diameter of capillaries, increases in diameter of capillaries, or we can say dilation in capillaries, increases the blood flow. Increases in blood flow help in increase in movement of cells. These cells go on the place where wound is present and fight against the bacteria that are trying to enter through wound. Third is increased vascular permeability. Through the movement of diapedasis, these cells squeeze out from the capillary to the wounded area. This is also known as leakness from blood vessels and recruitment of cells and fluid edema. You have always seen that when we have a skin damage, we have a tissue damage, or we have a wound, the region swells up. Why this swellings occur? First swellings come, then there is redness. Swelling is due to engorgement of capillaries. The blood capillaries move towards the upper surface. In that condition, the blood vessels, when they move on above surface where the wound is, there is flow of blood, and that flow of blood recruits different kind of cells to fight against the bacteria invading inside to wound. Extravasation of phagocytes, recruitment of leukocytes, chemotactic effect. Here we can see C3A and C5A. This is the complement protein part. N-formyl peptide. Action on blood vessels, intravascular adhesion molecules, ICAMs also function in recruiting different kind of cells. Then the final function is tissue repair through fibrin or clotting. There are certain clotting factors which works in the serum of blood, and they repair the damaged part. When we have any kind of wound, when we have any kind of damage, the blood squeeze out. In that condition, if we don't have proper clotting factors or we don't have fibrin, then clot will not form. So immune system works from the very beginning when it recruits the cells to fight against till it repairs the damaged area through clotting of the blood. Here you can see through this diagram. Here's something that damaged the tissue, and with this bacteria enters in our skin. After skin damage, when bacteria enters, there is tissue damage, cause release of vasoactive chemotactic factors that trigger a local increase in blood flow and capillary permeability. Second is permeable capillary allow an influx of fluid and cells. Here you can see that this is the blood capillary. Blood capillary becomes dilated, means it enlarges itself and moves towards the infected area or damaged area. After that, capillaries are made of very thin layer of cells. In these thin layer of cells, there are some phagocytic migrants that move towards the wounded area through the action of diapedasis. Here you can see this yellow cell is certain phagocytic cells like macrophage or monocyte. It is moving outside this capillary. This process is known as squeezing out, or the movement is diapedasis movement. Now the macrophage come in this area and now going to attack on bacteria through the action of phagocytes and antibacterial exudate and destroy the bacteria. So these are the 4 attributes of your innate immune response or innate immunity. Here I have completed this topic. Hope you are satisfied. Thank you.